Hello, I'm Szilveszter Juhos. I'm a bioinformatician at NGI, and I will speak about the cancer pipeline. That is a sort of collaboration between SciLife Lab, NGI, NBIS, and Karolinska. It is following practically, the library preparation what is usually done for whole genome sequences at NGI, and it is specialized for tumor normal pairs, so that we are not doing tumor-only cancer samples, and we are looking for somatic mutations, expecting low allele frequencies in a heterogeneous sample, and we are also providing variant codes for germline variants, and the software is designed in a way that you can add later relapse samples, and usually the expected coverage is for 30x and 90x, 30x for the normal and 90x for the tumor sample, but we are doing 45 coverage samples as well, and we are working on the whole exome and cancer panel methods as well. The beginning of the analysis is very similar to what we are doing for WGS, so that doing GATK best practices, and that is the most resource-intensive part of the whole analysis, eating most of the CPU time and disk space. And for this job, we are usually using Irma and Bianca because these are the UpMax clusters that are set up for sensitive data. We can use the old GRCH37 reference, but right now you can use the 38 as well. That is the latest human reference. And instead of using a single variant caller, we are applying a set of variant callers for SNPs and indels, and also for structural variants. Manta is to get quickly the rearrangement, and ASCAT is mostly for CNVs and also estimating sample heterogeneity and ploidity, or ploidy. And besides these variant callers that are specialized for somatic calling, we are also using a haplotype caller to get the germline calls and do some QC. So that people who are interested in cancer do know that the samples that we are usually getting are pretty heterogeneous, containing different subclones, and not that straightforward to analyze as for usual blood samples, normal samples. And after getting the results, we have to filter out variants. And this filtering is more or less a black magic, and we are trying to apply only really the basic filtering, so that by the end of the day we are not throwing away that much real variants, at least hoping. Also, the users will get not only the raw VCF files, but annotated files by SNP effect and variant effect predictor. This workflow is relatively new, and we tested it on synthetic datasets. Synthetic datasets are good because we know what to expect, but we are also working on test it on real data, so that we have public data that is available and contains a golden set of variants. And in the moment we are running all the analysis to optimize parameters. For example, what is the optimal coverage, what are the optimal filtering parameters. Besides these projects, the cancer pipeline already processed about 100 pairs for 5 different research groups. And the important thing is what are the files that are delivered to the users. Similarly to the whole genome samples we are providing the realigned BAM files with the recalibration table. It means that if you want to recalibrate it again, you have to do it yourself, but it is saving a lot of space. We are providing all the raw VCFs that are generated by the variant callers, and the filtered variant calls with annotations. What else? As I mentioned before, there is the software ASCAD that can estimate the sample heterogeneity. For example, so I got this slide from the ASCAD web page, and you can see that there are quite a lot of questions, and so that you will get an answer for all these questions, at least some estimate. This is a complex scenario, and you will get a complex answer, but at least pretty informative to know where are the chromosome gains or loss of heterozygosity and so on. And also there will be an average heterogeneity that you can get so that to estimate how many cancer cells are in your tissue. Besides this, we are also providing, similar to the whole genome sequencing experiments, the multi-QC reports. It is actually pretty long, so that I really just presenting some examples that you can find on the multi-QC website. This slide is probably the most important part from our point of view, showing the resources that you need. So usual processing for a single sample is 4 or 5 days for a lower coverage, or a whole week for a higher coverage sample. And the variant callers are about 10 hours for each variant caller. Some are shorter, some are longer, but the main thing is that the storage, that if you have about 300 gigabytes of raw data, raw FASTQ, by the end of the day, you will get about 7 or sometimes in a whole 700 or sometimes a terabyte of final data per pairs. And by the end of the day, you have to think about the storage of this data, because usually these are much bigger than normal whole genome sequencing samples. And NGI cannot really provide storage space. So we are users of UPMAX, as other users are also using UPMAX resources. So it is really good to think in advance about storage, especially for cancer samples. So for example, if you have 10 samples, 10 pairs, you will have to have at least 10 terabytes of disk storage for your samples. And this is the final slide, just showing all the contributors. You can see that it is not only NGI people, but many contributors from other sides of Uppsala or Stockholm. And that's all.